next speaker is going to be Professor Rebecca Hallett from the University of Guelph, talking about biology of switchgrass Gall Midge and its impact on switchgrass in Ontario.
Hi, it's my pleasure to be here today. I'm Rebecca Hallett. I am presenting this work on behalf of Clarissa Capco, who is a master's student with me. Clarissa, I think, is online, but she went home to BC, where she is madly writing up her thesis to come back and defend at the end of April. So I get the pleasure of presenting to you today. For those of you who aren't, oh, that's a shame that bar is going to cut off those headings, which is where some of the key messaging is. Anyway, if you're not familiar yet with the switchgrass Gall Midge,
push it that one stood down.
So the switchgrass Gall Midge is a small little cecidomyid fly that was first discovered in 2008 in South Dakota. Some studies there found that it caused nearly 100% seed loss and a 35% reduction in biomass per infested tiller. So if that's if you've got a lot of infested tillers, you could have really significant yield losses. This midge and its parasitoids were first found in southern Ontario in 2020 by Omapha, so including Mahendra and Hannah Fraser. And we've subsequently found it across Ontario in all biomass producing regions. So the larvae develop in the top node of the tiller, and the pupae stay within that top node, and then the adults will emerge. And here you can see the impact on infestation on the seed head. So this greatly reduced seed head that's produced.
Just click on the screen if it's not going. It should give you control.
There we go. Okay, so when we started this study, there were only 2 papers that had been published on switchgrass midge. Both of them from South Dakota, and they had relatively limited information about the life history and development of this midge. They reported having 2 larval instars that were sort of around 2 millimeters in size for their early, around 5 for the late instar. And then they reported seeing the pupae in adults, but they made no mention of eggs. So we had no description of eggs, what they look like, or where they're found. And so this just summarizes the phenological information. So the presence of different life stages over the season for the South Dakota site. I'm going to show you a number of figures like this. They all have the life stage at the side, and so the bars across show when that life stage was present. And then these black lines in the box means that those are life stages that were found in the previous year's growth, so in the overwintered tillers. And you can see that the information from South Dakota was rather incomplete in terms of the life history of switchgrass Gall Midge, but they did find here that they found 2 distinct times when they found the pupae in adults in the field. And so that suggested that there might be 2 generations of this midge in South Dakota. Although I'll note that the second generation in July was only observed in a very early maturing variety of switchgrass. And so it may be that the development of the midge is tied to the development of its host plant. So when Clarissa set out on her master's program, she had 3 objectives. First, to determine the phenology of switchgrass midge, so including looking at diapause termination and initiation. So just for clarity, phenology is the timing of each life stage throughout the year. Diapause is a period of pause development that is influenced by environmental conditions. And so in Canada, diapause usually is the way that insects survive the winter. Her second objective was to describe the parasitoid complex. So these wasps that parasitize switchgrass midge and use it as a host. And then to determine field infestation levels and the impacts of switchgrass midge in commercial switchgrass fields. So we did sampling to determine the phenology at multiple field sites over a 5-year period. Each week, the students would go out to the field, collect large numbers of tillers, bring them back to the lab, dissect every one of them, looking for the different life stages and recording what they found and when. So I'm going to take you through each of the life stages and what we've learned about it and when they occur. We were able to determine that there are 3 distinct larval stages that occur. So remember in South Dakota, they mentioned 2. So we found a much smaller early instar. We're not sure if they actually have 3 larval stages, but because we haven't done those precise studies, but it would appear so. And then they all develop in the top node of the tiller. The pupae stay there for overwintering. Sorry, they overwinter as larvae. So this is what we found then in terms of the presence of the larval instars. The late larvae overwinter in those tillers. They're present there from January to May. Then they begin to develop into pupae. And then these boxes show the weeks that the life stage was the most abundant during the season. So we found that pupae were most abundant in early to mid-June. And so based on that and the occurrence of these very little larval instars towards the end of June, that the adults ought to be present in June and laying eggs then. So then the early instar larvae are developing from June, most abundant towards the beginning of August, and then proceeding through the subsequent stages, the middle instar, the late instar, and staying in that late instar stage over the winter. I'll just say we've been unable to find larvae in vegetative tillers. The damage symptoms are only present in reproductive. You can only see the damage symptoms when the tiller is in the reproductive stage and it's affecting the inflorescence. We have dissected many vegetative tillers and haven't found any of those little instar larvae yet, but presumably they're there. So then in terms of determining when the adults are around, we've tried 3 different methods to try to capture adults and determine their timing. In 2023, we put these white cages out, filled them with cut overwintered tillers. So they should have those late instar larvae in them that are going to pupate and then emerge as adults. Yellow sticky traps inside those cages to capture the adults. So in 2023, doing this, we caught 2 adults. The next year, we built these large black mesh cages or black fabric cages, and they were either over standing switchgrass, which is why they're so tall, or filled with cut switchgrass overwintered tillers. And then the only light was at the very top of the cage, and so the adult should move towards the light, get captured in a collecting bottle there. And doing this, we caught 5 adults. So in 2025, Clarissa went out there with her sweep net 3 times a week to every field at the times of day when we expected, based on her lab studies, when the adults should be active, did sweep net sampling through the fields and found 0 adults. She captured 0 adults that way. So we had a grand total of 7 adults captured over 3 years. Whoops. Let's go back a little. Okay, and so this is what we know now about the adults. 6 of those 7 adults were found here in early June. 1 was found towards the end of June. And so that sort of confirms what we were expecting, but we still need to know a lot more about when the adults are around, about their biology and behavior, and how to detect them better. We need a better method of detecting them, because if a control method is ever needed, it would likely be targeted at the adults. There we go. So Clarissa also did some observational studies in the lab to look at reproductive or mating behaviors to try to determine, we were thinking that there was likely a female sex pheromone, like there is for many other cyssidid gall midges, that we could use in a trap to attract the males, and that would give us an idea of when adults were active and when egg laying was occurring. And we didn't find any reproductive or any sort of calling behaviors in females, but Clarissa observed a very cool thing. At least if you're an entomologist, it's very cool. And the first time that this has been seen with male midges in this family. So I want you to watch this male midge here. Normally his abdomen is rather gray, but here his abdomen is already looking quite orange. And then this is the female. She's on the side of the vial. She's going to walk up past him, and he's sitting on a pipe cleaner. So just for scale, that's how large they are. Okay, so watch that male. He's gonna, as she begins to go up, he's going to extend his abdomen, flutter his wings and legs. And then after she goes, he'll stop again. And so that is sort of behavioral evidence that the males are producing possibly a pheromone that might be attractive to the females. It could be that it's just the visual cue and the female has to be close by and see that bright orange abdomen, but we're thinking that the males are probably producing a sex pheromone. And so I, for those of you who were here last year, I gave a little bit of an update on some of the work we were doing with collaborators in the UK to identify the pheromone. And at that point I was saying, we can't find a female sex pheromone. Well, now we're looking at the males. My collaborators there feel that they have found a compound that only males produce, and they produce it in quite large quantities. And so in the spring, when we begin to get, we can collect pupae, bring them into the lab, and merge them and do some bioassays to determine whether there's behavioral response towards that compound. And if there is, maybe that becomes useful in monitoring the adults. So maybe more on that in another year. Okay, so the final life stage that we needed to figure out what they look like and where they're found are the eggs. Clarissa first found eggs. They're orange and very like long and cylindrical. On a potted plant in the lab, switchgrass plant that she put some midges in with, and then discovered that they laid some eggs on the youngest leaf on that plant. So that but we didn't really know in the field, we hadn't found any in the field. And so this past summer, Clarissa developed these oviposition cages. So these are cylindrical mesh cages that were designed to go over a vegetative tiller in the field. So there's one switchgrass tiller inside there, vegetative stage. She took pairs of adults out, male and female pairs, put them in each of 33 cages, and then went back and looked for oviposition. And so from that, very difficult to see, but there is a little egg mass right there, little orange stripe there. And so she was able to find 6 egg masses in this way, and that all of them were on that very youngest leaf of the plant. And so that let us know where we should be looking for these things. And so kind of armed with that knowledge, some more intensive sampling and focusing on looking at that youngest part of the leaf, but we only found one additional egg mass, like sort of loose, free in the field in July. But at least we have now found eggs in the field. Okay, so in terms of what we know at this point, South Dakota is at the top, Ontario at the bottom. If you remember, South Dakota felt that they had 2 generations occurring. There's no evidence of 2 generations of the midge in Ontario. It looks like we just have 1. We've confirmed that they overwinter in that late instar larval stage. We know that adults and eggs are present in June, July. And so that sort of may set the, provides us with some understanding of this midge if we do need to develop management practices. Clarissa did some studies on diapause initiation and termination. I'm going to skip over The initiation of diapause stuff in the fall for timing, but just will say that they seem to enter diapause, so stop their development in sort of October, November. But there's some evidence that that is affected by temperature, and so under future conditions, we may see additional generations develop. I'm going to talk a little bit about their development in the spring. So when diapause is terminated and the larvae start to develop again, because it's important for us to understand, you know, when they're becoming active in the spring, that might be when we need to target control methods. So overwintering larvae were brought into the lab and placed in different temperatures to look at their development rate and then try to predict the lowest temperature at which they would develop. And then Clarissa ran various models to look at that data, which are shown here. And that helped us determine that the minimum temperature at which they begin to develop in the spring is between 11.9 and 14.5 degrees Celsius. And so if you can imagine, if you extend these lines, these three lines down to the axis where no development occurs, those would fall between 11.9 and 14.5 degrees Celsius. So in Ontario, that's late April to May. The larvae begin to develop again and will become after that. That information is useful in developing predictive models for when the adults might emerge, so degree day modeling. Again, we want to know when that stage is coming to infest the switchgrass in the current year, and that might be where management practices or interventions are directed. So I mentioned that parasitoids of switchgrass midge were found in Ontario. We've been able to identify 4 different species of parasitoids that occur here. 3 of them we've been able to identify to the species level. A 4th one, this Quadristicus species, we haven't been able to figure out what species within that genus it belongs to. So in addition to knowing their names and having morphological descriptions of these, Clarissa also developed DNA barcode libraries, and those are publicly available. And so now somebody could take parasitoids that they find in the field, take some of the tissue, extract the DNA, do the sequencing, and compare them to the barcodes and determine what species is present here. So that's a quicker way of being able to identify them if you're not a taxonomist who knows how to look at these tiny little creatures and figure out what species they are. We found that these parasitoids are pretty abundant. Over 50%, 50 to 58% of tillers contain at least one parasitoid in these 2 years of sampling. And here, this is the middle graph shows the pupae, and we've routinely found over 100 pupae each week in the tillers that were collected. So that's in 60 tillers. And then you can see from the life stage timing that the adults of those parasitoids are emerging late June, July, around the same time that the switchgrass midge adults are emerging and laying their eggs. So they seem to be well synchronized with their host. And that's good in a biological control agent. If they're going to have any impact really on the population of the pest, then that synchrony is important. And so any practices for management of the pests that are developed should take into account the fact that we have these biocontrol agents out there in the field already, and do what we can to conserve them because they're helping us out. Okay, final objective of Clarissa's thesis was to look at infestation rates and yield impact in the field. So this shows 2 sites where we did quadrat sampling in the late fall. And from these 0.75 meter square quadrats, every single tiller was collected, separated out as an infested or an uninfested tiller. And at different distances from the edge of the field. So each field was divided into 4 zones based on distance from the edge of the field. And then within each of those zones, there were 4 quadrats that were collected. And so you can see in both years at both sites, almost all of the locations that were sampled had infested tillers in them. And also that so it's across the whole field. The midge is not restricted to the edges of the fields, which is common in some other midges that I've worked on. And then the percent of those tillers in the quadrats that were infested was higher at both sites in 2024 than in 2025. But around about 2% or less, except at site A in 2024, where we found 8.9% of those tillers were infested. So in 2024, the students then took those tillers that were infested or uninfested, separated the seeds off of the tillers, and then put all the vegetative parts together, and the seeds were separately, and they were both dried and weighed so that we could look at the impact on the different parts of the plant. And so this first chart on the left is seed weight at both site A and B, and the infested is always in the shaded boxes, and uninfested is the clear white boxes. And seed weight at site A was found to have been decreased 70% in those infested tillers. So fair impact on seed weight. At the other site, at site B, there was a numerical decrease in seed weight, but it wasn't statistically significant. Tiller weight, on the other hand, was found at both sites to have increased significantly with infestation. So something causing an increase in tiller weight with the switchgrass midge. In 2025, we did whole weights only, so we didn't separate off the seeds. It's incredible how time-consuming and laborious that is on all these thousands of tillers to separate the seeds out. So we did whole tiller weights in 2025. In site A, we found again that like whole tiller weight had increased. Infested tillers are heavier than uninfested tillers. The same wasn't found at site B, but there was fewer or very variable numbers of infested tillers in the plots here, and so there's no significant difference between the infested and uninfested, but it's numerically a decrease in tiller weight. Okay, so percent infestation is low though. At site A, we had about 2% in 2023. It went up to that 8.9%. And then back down again in 2025. We were concerned last year that this increase might keep on going, and that with the buildup of populations in a site, it doesn't appear that occurred, but it does indicate that there may be environmental conditions in particular years that allow very high population increases to occur with the midge, and you get these higher rates of infestation. But at site B, the infestation rates were pretty steady at about 1.5%. So our overall infestation rates in 2025 was 1.6%. So now if we take that to the field level, if you have 1.6% of your tillers across your whole field are infested, there's going to be a 27%, those infested tillers are 27% heavier. That would give you across the whole field just a 0.44%, less than 0.5% increase in tiller weight. And each tiller on average has a 50% decrease in seed weight. So across the whole field, that's 0.84% or nearly 1% decrease in seed yield. So at this sort of infestation level, it's probably of no concern. But if you think about to 2024 with that 8.99% of infestation, then, you know, at 9-10%, you're looking at 5% seed loss. And so I'm assuming that for a seed producer is beginning to get to an economic level of damage. So something to keep track of. So in conclusion, then, what we know about switchgrass midge, we can only see injury at the reproductive stage. It's the vegetative tillers don't show any signs of infestation. Has 1 generation per year, but it's possible that 2 could occur under future climates. The percent of switchgrass midge infestation is low right now, but it's variable, and so we should keep an eye on that. And then only seed production is negatively impacted by switchgrass midge. So if you're producing tillers for biomass rather than for sale of seed, then it's probably not a significant issue at the moment. But populations could change in the future, and the parasitoids are abundant and here to help us out. I'm gonna yeah, so if you're interested out there in your field, or if you're a crop manager, then things that you might want to do are to monitor infestation of switchgrass midge in August or September, when you can see those bright orange late instar larvae in the tillers. Go out, sample, see what you've got, get an idea from year to year of what your populations are doing. In stands with high levels of infestation, you might want to be selling your tillers for biomass rather than instead of seed, to minimize economic losses. In terms of trying to reduce populations from year to year, removing overwintered tillers before May, when those larvae have begun to develop into pupae, and the pupae are going to become adults, it's a good idea to make sure you've got all of your tillers from the previous year off the field. And then you could confirm the presence of switchgrass midge parasitoids by looking at those overwintered tillers in May, because they look quite different than the switchgrass midge do. I'm going to skip over future research, except to say we will keep trying to work on this male sex pheromone to see if we can develop a good monitoring method. And then there are some other questions to look at in terms of reducing populations and what switchgrass midge might be doing in different varieties, whether or not this effect of quicker maturation in those short varieties is real and occurring here as well. So those are things that could be looked at in the future. We'll stop there. Thanks very much.